I'm Owen Schwartz. I'm head of the biological imaging facility here, which is the core imaging facility for NIAID. What we do is we provide cutting-edge microscopy and imaging equipment for the whole DIR, Division of Intramural Research. And so the individual investigators prepare their samples, come down, talk to us, ask what the appropriate preparation should be, what would be best for imaging, tell us a bit about the problem they're trying to solve, and ask what the best approach would be. We recommend which instrument would best capture the kind of data they want, and then actually help them to operate the instruments. We're sitting in front of our newest imaging piece of equipment, a STED, or Stimulated Emission Depletion Microscope. It's a form of ultra-resolution microscope that allows us to see things that are almost an order of magnitude smaller than the best microscopes were a couple of years ago. On the left here we have a confocal image, one of our normal high-performance microscope images of chromosomes. And when we switch on the STED, or the emission depletion microscope, the resolution's enhanced and you can actually see that there are two chromosomes here. It's double-stranded. And this is one of the first times we could actually see this with visible light. The way it actually works are, there are two laser beams that are turned on simultaneously. The conventional one, which would be blue or green or red, visible light would come through the standard confocal scanner, go into the microscope and excite the fluorescence, make the sample glow. Superimposed on that is an infrared beam that comes through this component here. The infrared beam is shaped into a donut, and it serves to actually turn off the fluorescence or deplete it in the regions of the donut. And in the hole in the center, what's left is purely the in-focus light at a higher resolution than we had before. We also have a laser microcapture system, which allows you to cut out small pieces of tissue to do analysis, compare one cell to another, infected versus uninfected. People do a lot of genetic work, PCR and things based on small samples that way. We have microscopes for imaging whole animals, intravital imaging or multiphoton, which allows you to focus deep into tissue to watch the immune system in an intact, anesthetized mouse. One of the best days was when we got an entirely new microscope system working. It was my idea, and it took about 2 years to build and test, but it gave us 4 colors of intravital or of live animal imaging simultaneously. And seeing the first images come off of that just proved that everything we did for years and years really was worth it. I also recall sitting in our BSL-3, our high containment lab, and watching a movie playback of a live polio infection over time. It took about 8 hours. And seeing that, the fact that we could capture that sort of hazardous material but in live, real-time imaging was really, really satisfying.